Welcome to Study Loop.
For this question, we're going to look at the mitochondria and its role in ATP production under the stress of either carbon monoxide or cyanide, or under the stress of a toxin called rotenone. So if you recall the structure of the membranous protein chain inside the mitochondria, you have, of course, the lipid bilayer creating that membrane, and you're going to have a series of proteins embedded inside that membranous layer. For normal ATP production, you're going to have the movement of an electron through this series of proteins, and it's captured by oxygen to create water. And through this process, you're going to end up pumping protons into that matrix space of the intermembranous layer. And those protons are going to play a role in ATP generation through the protein called ATP synthase. So here you just have the protons traveling back into the insides of the mitochondria, and that will generate that ATP. So for that carbon monoxide or cyanide toxin inside the system, it's going to inhibit one of the protein carriers between this series of proteins that you may know as coenzyme Q. So coenzyme Q, or ubiquinone as it can also be called, is responsible for carrying this electron between your first protein in that electron chain over to that third protein. So when that occurs, you're no longer going to get a significant movement of those hydrogen ions into that intramembranous space, which is going to decrease the amount of ATP. And as you have this decrease of hydrogens compiling in the intramembranous space, and you have these sort of pileup of electrons as they can't be carried over from protein 1 to protein 3, you're also going to get a buildup of NADH because it can't deposit its electrons through the system anymore. So it's going to remain as NADH and be unable to be converted over to its NAD product. So the results of carbon monoxide or cyanide poisoning are going to be the accumulation of NADH. You're going to have a decrease in the protons in that intramembranous space because you're no longer going to be able to move that electron all the way through the system and pump those electrons out. And you're going to get an overall decrease in ATP production because you're not producing this proton gradient, which is going to generate ATP. For part B, you have rotenone, which is going to be a structure that is going to prevent the movement of electrons from protein number 1. So here, scientists have figured out the role of a substance called methylene blue. So methylene blue is going to be able to take the electron from NADH, and it's going to contribute it over to its oxygen counterpart. So this is going to prevent this buildup of NADH, and it's going to allow this electron to sort of move through the system and at least generate some degree of ATP in the final, as it's finally captured in oxygen and water to be excreted in the body. So the use of methylene blue is going to be able to create another pathway for the movement of that electron when rotenone is going to block the original function and movement of electrons through the electron transport chain. So here, methylene blue is just going to be a sort of intermediary substitute. for the electron transport chain. And that would allow the individual to produce that ATP and to reverse some effects of Alzheimer's and increase their body's metabolism.